Marampally, e-learning.
Hello everyone, I am Ashwati Vijay, Assistant Professor, Department of Biosciences, MES Marampally. Today, I am going to deliver a lecture on the topic, the beta oxidation of fatty acids. The oxidation of foodstuffs is the basic step for energy release in any cells. Apart from carbohydrates, fats also serve as great energy source for most of the cells. The beta oxidation is a catabolic process where the fatty acid are oxidized to acetyl-CoA to produce ATP. So now let's take a look into detail about the topic.
Cells can obtain fatty acid as fuel from two sources. One is through the fats consumed in the diet and the other source through the fats stored in adipocytes as lipid droplets. The fat obtained via these two sources undergo digestion to release fatty acids. The oxidation of long-chain fatty acids to acetyl-CoA is a central energy-yielding pathway in many organisms and tissues. The product of oxidation of fatty acids is acetyl-CoA, and this oxidation process forms a central energy-yielding pathway in most of the organisms and tissues. In mammalian heart and liver, fatty acid oxidation provides as much as 80% of the total energy needs. However, brain, erythrocytes and adrenal medulla cannot utilize fatty acids for energy production. The enzymes of fatty acid oxidation in animal cells are located in the mitochondrial matrix. So the site of fatty acid oxidation in animal cells is the mitochondrial matrix. Mitochondrial oxidation of fatty acids takes place in 3 stages. So, the oxidation of fatty acids involves 3 steps. First one, activation of fatty acids occurring in the cytosol. Second step, transport of fatty acids into mitochondria. And the third and final step, beta oxidation in the mitochondrial matrix. So, the 3 steps are first one, activation, then transport, and finally beta oxidation. Fatty acids undergo oxidative removal of successive 2 carbon units in the form of acetyl-CoA at the 3rd carbon or carbon at the beta position. Hence, this process is called as beta oxidation. In beta oxidation, the fatty acids undergo oxidative removal of successive 2 carbon units in the form of acetyl-CoA at the 3rd carbon atom or carbon at the beta position. So we can look into detail about the first step, that is fatty acid activation. Before fatty acids can be oxidized, they must be primed or activated by an ATP dependent reaction to form fatty acyl-CoA. This activation process is catalyzed by the enzyme fatty acyl-CoA synthetase associated with the outer mitochondrial membrane. The reaction occurs in 2 steps and involves the formation of a fatty acyl adenylate intermediate. Fatty acid reacts with ATP to form fatty acyl adenylate or acyl adenylate, which then combines with coenzyme A to produce fatty acyl-CoA. So the overall reaction can be represented as fatty acid plus coenzyme A plus ATP in presence of the enzyme fatty acyl-CoA synthetase form fatty acyl-CoA plus AMP plus PPI. second stage of fatty acid oxidation, that is transport across the mitochondrial membrane. The fatty acids with chain length of 12 or fewer carbons enter mitochondria without the help of membrane transporters. But those fatty acids with 14 or more carbons and which constitute the majority of free fatty acid obtained in the diet or released from adipose tissues cannot directly pass through the mitochondrial membranes, and they require a transport mechanism which is known as the Carnitine shuttle. So most of the free fatty acids enter the mitochondria matrix by the Carnitine shuttle. The steps in Carnitine shuttle are: first one, the acyl group of acetosolic acyl-CoA is transferred to carnitine catalyzed by the enzyme carnitine acyl transferase 1, thereby releasing the coenzyme A to its cytosolic pool. Second step, the resulting acyl carnitine is transported into the mitochondrial matrix by the transport system. Then, third step, the acyl group is transferred to a coenzyme A molecule from the mitochondrial pool by carnitine acyl transferase 2. And finally, the product carnitine is returned to cytosol. This is a diagrammatic representation of the carnitine shuttle. So fatty acid enters into mitochondria via the carnitine transporter. So in the diagram, cytosol, inner mitochondrial membrane and mitochondrial matrix are shown. In the cytosol, at first, acyl-CoA combines with carnitine by the enzyme carnitine acyl transferase 1 and form acyl-carnitine, and coenzyme A is released to the cytosol. Then acyl-carnitine with the help of carrier protein enters the mitochondrial matrix, and inside the matrix the acyl-carnitine then combines with coenzyme A catalyzed by enzyme carnitine acyl transferase 2, and fatty acyl or acyl-CoA is formed, and the carnitine is shuttled back to cytosol in order to continue the shuttle. Then coming to beta oxidation. The beta oxidation of saturated fatty acid has 4 basic steps. So 4 steps or 4 reactions are involved in beta oxidation. In 1904, Franz Noot proposed that the breakdown of fatty acids occurs by a mechanism known as beta oxidation, that is, oxidation of fatty acids on the beta carbon atom. Step 1, oxidation catalyzed by FAD-linked dehydrogenase. In the first step, dehydrogenation of fatty acyl-CoA occurs, producing a double bond between the carbon atoms at the second and third position, and the product yield is a trans delta 2 enoyl CoA. Here the symbol 2 designates the position of the double bond. And the reaction is catalyzed by the flavo enzyme acyl-CoA dehydrogenase. Since it has a fatty acids coenzyme, it is known as the flavo enzyme. Then second step, hydration. Hydration of the double bond by enoyl CoA hydratase form beta hydroxy acyl CoA. Then third step, second oxidation catalyzed by NAD plus dependent dehydrogenase. NAD plus dependent dehydrogenation of beta hydroxy acyl CoA forms keto acyl CoA, and the enzyme involved is beta hydroxy acyl CoA dehydrogenase. Step 4, cleavage or thiolytic cleavage. Thiolysis reaction with coenzyme A catalyzed by thiolase to form acetyl-CoA and a new acyl-CoA containing 2 carbon atoms less than the original one, and this new acyl-CoA re-enters the beta oxidation cycle. The process or the 4 reaction continue till the fatty acid is completely oxidized. In case of palmitic acid or palmitoyl-CoA, palmitoyl-CoA undergoes 7 cycles of beta oxidation to yield 8-acetyl-CoA, and the overall reaction for the oxidation of palmitoyl-CoA can be represented as palmitoyl-CoA plus 7 coenzyme A plus 7 FAD plus 7 NAD plus 7 water molecules form 8-acetyl-CoA plus 7 FADH2 plus 7 NADH plus 7H plus. This is the structural representation of the 4 steps of beta oxidation. So here palmitoyl-CoA or palmitic acid is considered. At first, oxidation catalyzed by the enzyme acyl-CoA dehydrogenase. It contains FAD acids, coenzyme or prosthetic group. So FAD accepts 2 hydrogen atoms and forms FADH2, and the resulting product is trans delta to enoyl CoA. RCH2 CH double bond CH C double bond O SCOA. Then hydration occurs in the second step, catalyzed by enoyl CoA hydratase. So the product becomes RCH2 COHH CH2 C double bond O SCOA, that is beta hydroxy acyl CoA. Then the third step is again or second oxidation, catalyzed by beta hydroxyacyl CoA dehydrogenase, which has NAD plus acids coenzyme. Here also NAD plus accept 2 hydrogen atoms and form NADH plus H plus, and the resulting product is RCH2 C double bond O CH2 C double bond O SCOA, that is beta ketoacyl CoA. Then thiolase enzyme catalyzes the final step, that is thiolytic cleavage, and acetyl-CoA is formed, and a new acyl-CoA containing 2 carbon atoms less than the original fatty acyl-CoA is also formed. So RCH2 C double bond O SCOA with 14 carbon atoms and a molecule of acetyl-CoA is formed after one round of beta oxidation. Then this acyl-CoA, that is myristoyl-CoA, then again undergoes these 4 reactions of beta oxidation cycle till the fatty acid is completely oxidized. So in total, palmitic acid undergoes 7 cycles of beta oxidation to yield 8 molecules of acetyl-CoA. Coming to energetics of beta oxidation, that is the number of ATP molecules produced by beta oxidation. So here we are considering the beta oxidation of palmitic acids. So beta oxidation, 7 cycles, 7 FADH2 are formed, and this 7 FADH2 can enter electron transport chain or ETC, and 1 FADH2 gives 2 ATP. So from 7 FADH2, a total of 14 ATP can be obtained. Also 7 NADH are formed, which also enters the electron transport chain. Each NADH can liberate 3 ATP. So in total 21 ATP can be formed from 7 NADH. Then 8 acetyl-CoA are produced, which enter the citric acid cycle, and one or each acetyl-CoA provides 12 ATP. So in total 96 ATP can be formed from acetyl-CoA. So total energy produced from one molecule of palmityl-CoA is 131. We have utilized 2 ATP for activation. For the formation of fatty acyl-CoA, 2 ATPs are utilized. So the net yield or gain of ATPs from oxidation of one molecule of palmitate is 129. That's all about beta oxidation. Thank you everyone for listening.